On this week's OIS Retina podcast, we sit down with Patrik De Haes, MD. Following a prestigious career in eye care involving tenure as global head of immunology at Sandoz, now Novartis, Dr. De Haes currently serves as CEO of Oxurion, a biopharmaceutical company developing next-generation standard ophthalmic therapies.
Welcome back, everyone, to the OIS Retina podcast. Thank you for joining us. This is Firas Rahal. I'm a partner at Retina Vitreous Associates in Los Angeles, which is where I'm broadcasting from. And I'm a partner at Excite Ventures, which is centered in New York City. Today, I'm very happy to have my friend and a very important person in the field as our guest, Dr. Patrik De Haes of Oxurion. Patrik is the CEO and member of the board of directors of Oxurion. We're going to speak a lot about what Oxurion is doing and how he got there. Maybe we can start there, Patrik. I have a little bit of your history. You and I have met and talked and had some laughs before, but I was happy to learn a little bit about your previous history in the biomedical space. And it's long and it's very impressive. It looks like you had 25 years of experience in the global health industry. You were with Roche. Maybe you can tell us what you did there. And you're also with Novartis. Why don't you take a minute, tell us about your personal, professional history there and how that led you to the current position with Oxurion.
Well, thank you, Firas, and happy to be on the program. The 25 years experience, that was 10 years ago. So so it's about 35 now, because, after I studied medicine here in Leuven, Belgium, I got an offer, actually. I got an offer from what was called Sandoz, now Novartis, to become their global head of immunology, allergology, and infectious diseases. That was my first role, and my potential career in internal medicine, I kind of moved away, and we moved to Switzerland. From there, I got involved in the development of the first biotech product for Sandoz, which was a colony stimulating factor, GM-CSF, competitor to G-CSF, Neupogen. And I was the global project leader for that. And that drug got launched in the 90s. But then I got an offer to move to the States, to Minneapolis, to become CEO of a medtech company. A medtech company that was focused on diabetes pumps, insulin pumps. And we basically, it was a beginning market. Now it's a multi-billion market, but in those days, only two contenders. We sold that back in 2003 to Roche. And I became a manager of Roche for that business. And then, I got contacted basically to run this biotech company in Belgium, but it was basically a university spin-off involved in vascular diseases. And I moved it from vascular to ophthalmology, and right now we're a pure ophthalmology play. So that's the history for us.
Yeah, I know a little bit of the history of Oxurion, and I didn't know when it began and how you did start there. It's currently a public company, is that right?
Yeah, it's a public company since 2006, listed on the Brussels Stock Exchange.
How's that different for you? You've been involved, I'm sure, as you said, in smaller startup private companies, and this was, I'm sure, originally private and then became public. How's that different for your role as a CEO or any other upper level management position, the functioning?
Well, a private company, you have, let's say, next to the people that work for you, you have about 10 stakeholders. In a public company, you suddenly get 10,000 stakeholders. So it's a completely different ballgame. It's like a public company has to do a lot about perception. The perception. you don't have to deal with that too much in a private company, but let's say making sure that your 10,000 stakeholders are happy is quite a challenge and takes quite a bit of time of senior management in public companies.
Yeah, totally. That makes sense. Before we delve into the product line, which is obviously where we want to spend a lot of time, you're doing fascinating science and you're clinical now, and I want to get into that. Just a little bit more about the history. One, a personal history. You're a physician. You went to medical school. Did you come right out of medical school and go into the industry side, or did you do residency training? And either way, what led you down this career path at that early stage?
Yeah. So what happened, this is now early 80s. You graduate from medical school, and then, I did a pre-training for internal medicine. So at that stage, you go into, let's say, internal medicine. But in those days, you always had to do military service. Mm-hmm. It was still, 1 year of military service, which you can do basically when you come out of medical school or after your specialty. So what I decided was basically do the military service first. In the meantime, I was practicing medicine for the army, but also, let's say, in private practice. And let's say it was during that time that I got contacted by Sandoz, Novartis, to kind of get involved in that. And yeah, I had the choice, but it was a big gamble, of course. It's, or you go back to the university hospital to do your specialty education, if I may call it like that. But, I opted with my very small family to kind of make the jump, move to Switzerland and get into a completely different business. So, everybody in life makes a certain gamble. I made that gamble at that time, and I have not regretted for a minute.
Well, the gamble paid off for sure, but I'm sure, having gone through myself med school and residency and all the different decisions along the tree of choosing to do a fellowship or otherwise, ultimately I did a fellowship. There is risk assessment for yourself personally at each one of those steps. And there is a little gambling. And I give you credit for making the decision that you thought would make the most sense for you. And obviously in retrospect, it's paid off. So, congratulations for that. But I know that must have been a difficult decision, especially if you had a family at the time. And all the expectation that most people go to med school, come out and do clinical medicine, but a certain significant minority do not and are very happy with that, I think.
Yeah, of course, it's like your parents, they say, what are you going to do? You're going to sell drugs? I mean, you went through
medical school.
Yeah, I know you're going to sell drugs, because that's how they looked at that, join the pharmaceutical industry. And let's say, in those days, physicians rarely would make that move. Right now, it's very frequent. Yeah. But in those days, you must be pretty much out of your mind to make that move.
Yeah.
Maybe I still am, but
nobody
tells me.
When you're young, they say you're crazy. Then when you get gray hair like us, you're crazy like a fox. So you're now in the crazy like a fox status. So good for you. You accomplished. Great. And the other history I was interested in is more on the professional Oxurion side. The early phases of the company, you said, were not in ophthalmology. What was that history, briefly, if you can, and how did you decide to transition to ophthalmology in those earlier days?
So at the very, very basis is a drug called TPA, tissue plasminogen activator, which a local professor here at the university, he basically found TPA and licensed that to a small biotech called Genentech. This is now 1979.
Yeah.
Long time ago.
Yeah.
So of the licensing deal with Genentech, there was about $140 million that came back to Leuven. And part of that was basically invested in, let's say, the start of what is currently called Oxurion. The successor to that was, to TPA, was microplasmin. because microplasmin is also a clot buster. And what happened is that Mike Tracy in the Midwest, he found that it was plasmin that releases the vitreous from the retina. And we tested our molecule in the back of the eye, and we saw that microplasmin at the right dose was able to basically release the vitreous from the retina. And that's how basically this vascular product became an ophthalmology product. And then over time, we started moving the company from vascular to ophthalmology, and right now we're a pure play in ophthalmology. But that's how it started, with TPA, Mike Tracy, and then Jetrea, and now the new portfolio with diabetes products.
I remember the earlier days when Mike Tracy and the other pediatric retina surgeons were toying with this idea. And I was in New York at the time, early in my career, and it was brilliant. We knew it made a lot of sense. It just, it had perfect biological sense that you could enzymatically cleave the vitreous. And we had great hopes for a lot of other potential solutions from that, including early studies, even when I was here in Los Angeles, of using some type of vitreolysis to treat diabetic retinopathy and create the posterior vitreous detachment, which in some ways can be curative for the proliferative form or late non-proliferative form. It's fascinating. It never quite went to that level, and maybe it still will. I don't know. Is there still work really aggressively happening in this area of the cleavage of the vitreous for treatment of diabetic retinopathy? Do you know? Are you guys interested in that anymore?
No. What happened is that so we licensed the project Jetrea to Novartis, Alcon ex-US, and we ourselves launched the product in the US. But whether in the US, whether outside the US, it was never a commercial success.
Yeah.
So we decided already quite a few years ago to basically, pull the commercial plug, in terms of, we said, okay, we keep supporting this commercially, but there's really no traction, no pun intended there, but there's no traction for that product. And that's why, we decided together with the board to kind of invest that money in new molecules and build up a pipeline. And that was, I think, the more straightforward thing would have been, you keep supporting your commercial product and then you get a certain revenue. But by doing that, we thought we're going to basically take the future away from the company, because the future, we didn't think it was going to be Jetrea. The future needed to be new molecules. And in those days, we saw, let's say, more people in the wet AMD space active than in the DME space. And that's why we started to basically, focusing the research on the DME space about 5 years ago.
Great decision. Obviously, you came to that decision as a group, I'm sure, with a lot of thought and trepidation, and also gamble. There's a lot of money at stake there, and congratulations. Let's move into those products that I alluded to earlier and that you just alluded to. I'll start with the first one. The THR-149 is one of your principal products in the pipeline right now. I believe it is in clinical testing. It's a plasma kallikrein inhibitor, which I know something about, but not a lot. Maybe you can tell us about this product in any level of detail or broadly that you feel you'd like to discuss.
Yeah. So, we have been strong believers in the PCAL pathway, for many years. PCAL basically inhibits bradykinin, and that is a very known, let's say, product that causes edema in the back of the eye. We work together with a company called Bicycle Therapeutics. So our molecule is a biologic, bicyclic. It has two cyclic entities, And it's a very powerful inhibitor of that PCAL pathway. We even think or know that it's the strongest inhibitor for that pathway. So all the preclinical studies that we have done, and let's say our staff in preclinical is over 20 people. So we have a sizable number of preclinical scientists in-house. And the focus is on all those preclinical models in DME. And let's say everything led to the belief that the reduction of central retinal thickness and the improvement of BCVA, which is not always correlated in the humans in DME, we know, but that's what we wanted to see in the clinic. So we entered the clinic about, I would say, a year and a half ago. I can't exactly recall. And we did a phase 1 study, and we saw that on day 1, we already gained 4 letters. Day 14, we have 7.5 letters. And amazingly, day 30, we still had, let's say, between 6 and 7 letters gained with 1 injection. And based on that data, we said we're going to have to go as soon as possible with multiple dose. And that's the study we are currently recruiting in the US, in Germany, France, Spain, and a host of other European countries. And that's where we should have first results in the middle of next year. So we test 3 doses and we inject 3 times and then follow the patients up to 6 months. So that's currently the focus on this on that PCAL. We know that certain attempts have been made in this space, but we're not sure that the molecules that were tested were as powerful as the one we have.
So that answers a bit of the question I was going to ask you subsequently in a way. I'm familiar with the Calvista product that we were involved in one of their early trials. And is the main difference between the two is just strictly the molecules different? The mechanisms and the MOA are the same, but different molecule trying to achieve the same thing?
Yeah, we do believe that we have, let's say, the more powerful molecule. We only inject 50 microliters. So it's a pretty powerful molecule. That's why we do believe that, let's say, to make big conclusions out of the Calvista data for our molecule, I don't think that is fair. I know it's happening, but the world wasn't made to be fair. That's clear too. I hope that people are able to take an objective look at the data that we have and that we will come out with middle of next year.
All right. With regard to the clinical data is quite good, as you indicated. It's early, but I think people should absolutely have an open mind. The mechanism makes a lot of sense. With regard to this PCAL-kinin system, it's separate from anti-VEGF. My understanding is there's some overlap between the two branches of these systems. Would this be then, in your mind, clinically for non-responders to anti-VEGF? Would it have a role in adjunctive therapy? How do you view the positioning for this from this early stage?
Yeah, so we as a company focus on VEGF-independent mechanisms. I know there's many companies trying to optimize the VEGF pathway, but according to the literature, and as you know from clinical practice, there's about 40% of patients what we call suboptimally responding to anti-VEGF. And it's in that 40%, let's say, that we do believe that we want to put our bets. In that VEGF independent group, you have basically Roche with faricimab, and then you have the PCAL pathway and the integrin inhibition pathway. And we play 2 out of the 3 pathways. So that's where we strongly believe VEGF independent. So the positioning for the product is second line. So in the future, people will give basically generic anti-VEGF because they become generic, bigeneric. If, let's say, you don't have an optimal response on the VEGF pathway, right now people switch molecules, but we hope to be able to come up with a molecule where you basically take a completely other route and are able to put that second line. Now, you brought it up as an adjunct. We have developed a molecule where we link an anti-VEGF with a pCal inhibitor. You know, we are currently putting the focus on pCal alone to show what it can do on its own. But if pCal basically would address a certain part of the population and you could link that to an anti-VEGF, you potentially would have a strong molecule covering a broad range. But the first positioning is clearly second line anti-VEGF suboptimal responders.
I have to really congratulate you on how you say that, your clarity, how you phrase it, because you talk to a lot of people in our business. I do too. It's infrequent that I hear this kind of clarity about a new product and its positioning relative to the current thinking and the current excellent treatments. As an investor, I have said many times to startups, people looking for funding and so forth, that the VEGF drugs, despite the 40% that you correctly identify, is a very high bar. We've never had drugs this good that work this consistently for this long in such serious conditions. So constantly having people jump into that water to defeat and exceed this high bar, I think sometimes is a dangerous strategy. And even when they are, I think, hesitant, they're very hesitant to say that they're going to go down another path very distinctly like you have. You really should be congratulated for that. I think that's a really good thought process on how to position your product. There's no reason to think you wouldn't have a very vast and excellent marketplace if you're choosing from 40% of the deluge of DME patients. Was this something you had a lot of conversations about at your company in the beginning?
Yeah, but it's so evident that, let's say, you have everybody and his dog fighting on 60% and trying to improve that in a biogeneric environment. Whereas on the other hand, you have a medical need. Our idea was, where is the medical need? Where the medical need is, you need the molecules. And of course, what this brings, it brings more risk, but also more reward. But, I didn't come to biotech to kind of improve something that exists. We didn't do that with Jetrea, and we are not doing that with pCal, but somebody has to do it. Somebody has to be, somebody has to put the eye on OIS, right?
Yeah, right. Yeah, no, that's the innovation, is the new molecules. That really is the innovation, not just piling on to the already existing paradigm.
And that's why, I'm very grateful to have a very knowledgeable, but also supportive board to kind of take this high risk approach. Of course, the market as such in the COVID, is moving away from risk. That's also a reality. But I always believe, it's like somebody has to do the innovation, and that's somebody. We did it with Jetrea, and it's like we're gonna come back with the pCal, with the integrin inhibitor. But one of the things that we learned is that with Jetrea, we had one molecule in hand. With the DME space, we go for multiple shots on goal. And
let's talk about one of the other shots then. Sorry, I didn't mean
no,
to
please.
I want to give you a chance and speak about the integrin antagonist. So this is THR-687, if I'm not mistaken. It's an integrin antagonist. Maybe you can tell me what RGD stands for. I wasn't familiar with the term. Totally different mechanism of action, same patient population. Perhaps this is for 100% of DME patients. I'll let you take it from there. Fill in to us about these different ideas.
Yeah. So the the integrins, they were developed, let's say, in the oncology space and some other attempts. Let's put it like this. What our preclinical scientists, indicated is that, they have a very broad mechanism of action. They call that pan-RGD. It's like, I need a CSO to explain all that. But the thing is that, it works. You have anti-VEGF, and upstream and downstream from it, the integrin inhibitors work. So it's a very broad acting. So we thought, if this is very broad acting and covers anti-VEGF, but also the pathways around it, that could be an interesting molecule. And what we found is that we had, in preclinical models in wet AMD, very strong results. DME, very strong result. RVO. So, we see a wide range of activity with these molecules. And there's even Allegro, who is testing integrin inhibition in dry AMD. At this stage, we leave that discussion, on the side. But what we think is that if you have such a powerful molecule, and we start in DME, there we would go for actually first line. For the reason that covers the VEGF pathway as well. So it could be a potential to kind of have anti-VEGF plus, but the plus with very different pathways. That's for us is so attractive. And we start the phase one. What did we find in phase one? With one injection at the highest dose, we got basically like 4 letters on day 1. We got over 10 letters day 14, even 12 letters. Wow. And 3 months later, with one injection, we still had over 10 letters gain.
One injection, 3 months, still 10 letters.
Yeah.
That's fantastic.
So we felt that was quite impressive. And we plan to start the multiple dose. I mean, if with one injection you get to 10 letters, it's like if we do 3 injections, we should really, hit it out of the ball game, like you say that in the US. And that's why, it's like the trial can start in the second quarter of next year. But we are eagerly anticipating, let's say, also that start. And then we have these 2 molecules which are clearly differentiated from one another. One second line, the other first line. And that's why we call it our franchise. We want to develop a franchise in the DME space. And that's what we do in clinic.
With your second phase trial, the one you were talking about for this product, will you be giving, maybe it's designed already, I don't know. Is it going to be monthly injection? Are you planning to look at monthly versus 2 or 3 months? How do you foresee that?
The first part of the study will be monthly injections. And then we see, then we follow the patients for 3 months. So you give 3 injections and then you follow it for 3 months to see how we do. And then afterwards you do probably your 3 monthly injections. Basically on the market research, we and other people have done is that, and maybe you can confirm or disagree with it, but what we hear is that docs like to see their patients back every 3 to 4 months in diabetes.
Yeah. I think with DME, 3 months makes good sense. It becomes almost like the glaucoma model, and it makes a lot of sense in the ophthalmology space. 6 seems a little long for this condition. 3 to 4 is the sweet spot. I would agree. You've got you've got the consensus, and I'm sure a lot of us agree on that.
And that's our target with the molecules, to kind of come up with a 3 monthly dosing interval.
These are very exciting products, both of them. I love the DME space myself. I have a big practice in diabetic retinopathy. We've been overjoyed at the fact we've been in the last 15 years being able to improve these patients, and the anti-VEGF drugs have, revolutionized what we're doing also in, for beyond DME and proliferative diabetic retinopathy. Even though that's not the labeled indication, we're all using these products to control neovascularization, pre and post vitrectomy. They're fabulous. Do you foresee this drug in the use of advanced non-proliferative or proliferative in the same way? Is there an anti-neovascular component that you think about?
Yeah, I mean, the strongest element is the anti-neovascular activity of that molecule. So the moment we have solid data with the multiple dose, we're going to be in wet AMD as a speeding bullet.
Yeah, good for you. And there's there's still a lot of need there too. There's plenty of treatment non-responders or limited responders. We have a lot of room for improvement in both of these diseases. We only have a few minutes. I had a couple of more specific quick things. The products are great, really. It's a very impressive data set on both of those products. Congratulations. What other molecules might you be looking at down the road? Are they going to be in this same category, or are there other mechanisms you're interested in?
Well, a year and a half ago, we decided that, let's say, with the 2 shots on goal in DME, we're fine. There were also more people coming in, but we still felt that in the dry AMD space, you had the complement approach, but beyond the complement approach, there are some, but we feel that, let's say, we want to be a player in the non-complement approach of dry AMD. So we set up these preclinical models in-house. We work with some very good academic consultants, and we have come to some very interesting pathways, which at this stage, we cannot talk about. But we are preparing let's say, also multiple shots on goal approach in the dry AMD space. So that will be for next year's OIS podcast.
We'll have you back for sure. I enjoy talking with you anyway, but that would give us a great reason to hear from you. So you're thinking you might be opening up dialogue about this within the next year or so?
That's the goal. That's the goal.
Yeah.
It's like the dry AMD space. You have that big discussion. Preclinical models are less predictable for the clinic and so on and so forth. You have different kinds of these preclinical models. But we remain still of the opinion that, we want to see some preclinical models, positive data before we move to the clinic. But the pathways identified by our preclinical people and confirmed by, let's say, external academics are intriguing. And in the year to come, we want to find ways to block those pathways, so it won't be boring.
That's amazing. One final quick question. I saw, and you can answer to the extent you feel comfortable. I saw on your website a mention of Oncurious, another corporate structure, a partner or an affiliate. Can you tell us briefly what that is? Well,
our previous name was Thrombogenics. And Thrombogenics, we basically had ophthalmology and oncology products. We have basically put the ophthalmology products in Oxurion and the oncology products in Oncurious.
Okay.
So in oncology, I don't think your audience will be excited about the oncology portfolio. So
right.
It's also in a clinical stage in medulloblastoma in kids, but it's a different ballgame. So our focus is basically on ophthalmology. We're looking for a new management, separate management, because right now I have been running these two things. So we're looking for a separate management for Oncurious, and that will become a completely separate entity also financially in the year to come.
Brilliant. That's a clear answer. And yes, that would be outside of our skill set for sure. Patrik, thank you so much for coming on. Your clarity is always really appreciated by myself. I'm sure by the audience. Your answers are clear. Your direction of your company has been made very clear. You should be proud of what you're doing, and thank you for coming on. Really appreciate it.
My pleasure, Firas, and thank you.
Thanks for listening to this exciting conversation with Patrik De Haes, MD. For more great retina content, tune in next time. And if you haven't already, subscribe to the OIS Retina Podcast at ois.net/subscribe.